So welcome, Dr. Arnold.
Kirsten, thank you so much for the really nice introduction. And I really want to thank you and Sue for the chance to talk a little bit with your group tonight and just express my enthusiasm for what a wonderful society you all represent. Growing up here in Arizona, I love the native plants. I was seduced into the fungal world despite doing my graduate studies in a plant systematics lab. And so I'm hoping that I won't poach anyone from your group tonight into loving mycology too much. But that at the end, everyone will walk away having learned something, and I would love to answer questions just as we go along. So really, thanks. And as I was thinking about this presentation, I was thinking, one of the joys of being a university professor is helping college students at that tender age navigate the existential questions like, why are we here, right? And for me, why I'm here, I think, is easy. It's this joyous enthusiasm for the natural world around us. I wake up in the morning thinking about fungi, and I go to sleep at night thinking about them. And hopefully a few folks will too at the end of this presentation. So, what we'll be talking about tonight is how Arizona showcases this tremendous biodiversity of fungi. But I wanted to step back for just a moment and recognize that when we're thinking about questions in ecology and evolution, they often come together in the world of symbiosis. That is the living together of two unlike organisms. And perhaps the most widespread and spectacular symbioses that we get to see with some frequency are the symbioses between plants and fungi. So in a sense, I also feel like I'm doing the right mycological outreach to plant partners tonight with this little presentation. Now, why are fungi so cool? I mean, why are they not, right? There's so many great reasons. They range from everything from the cause of athlete's foot to the very reason that beer exists and bread rises. This is where we find our most important plant pathogens, but also keys to the capacity of plants to live on land. The sources of some of our most potent antibiotics, and so on and so on. So to be studying fungi is to be studying the confluence of all of these things. Now, of course, when we think about fungi, the vast majority of us will think about these charismatic macrofungi you see across the bottom, right? The beautiful boletes and earth stars, cup fungi and morels. My specialty is actually in the microfungi. These are the ones that get my heart beating especially quickly. Basically the fungi we might call molds. And occasionally I like to talk with my mom about how all of those years of schooling paid off, because now I can really know a lot about mold. She's not always super impressed, but hopefully we'll have a little time to talk about the microfungi, those with microscopic fruiting bodies, as well as these beautiful macrofungi today. But being a professor, I am going to start with a little bit of vocabulary. I promise that because this is wine time, as it were, we're not going to have too many key words here. But I just want to highlight how fungi are a distinctive lineage evolutionarily. They are neither plants nor animals, of course, nor are they bacteria. They're eukaryotes. They are part of our tree of life. And in fact, they're more closely related to animals than they are to plants. But a quirk of human nature has led them to be in the eye of botanists, because they often co-occur with plants and they don't tend to run around. So often they're studied by botanists, at least in historical terms. And these eukaryotes are super neat because they never photosynthesize. They can only eat nutrients out in the environment. They're heterotrophs. And they have cell walls that are made of chitin, essentially the same material that we find in exoskeletons of insects. And so when we look at the beautiful tree of life and we think about the major groups that excite so many of us, we have plants on the one side, and they're sort of mirror image over here in the fungi, closely associated with the animal lineages. Okay. And so when we think a little bit about fungal diversity, it's a really fun sort of debate among mycologists. And we know that there are somewhere between 72,000 and 100,000 species of fungi that have been described so far. And so what we mean is there's a specimen and a Latin name and some information about the organism. But the going estimates are that there's somewhere between about 1.5 million and 9 million species of fungi on the planet. So for a fungal biologist like me, that's pretty great job security, that difference between how many we know and how many are out there waiting to be found. And in fact, a lot of my research is really fundamental biodiversity research, discovering fungi for the first time. Now, just as we like to so often look at flowers or cones or other reproductive structures for plants, so too in fungi do we have our eye drawn to these beautiful life phases of the fruiting bodies. And so, of course, many fungi are known for the beautiful mushrooms that they form, but there are a diversity of growth forms that are all worth noting. In the fungi, we find not only mushrooms of many, many different kinds, but also truffles, rusts and smuts, yeasts and morels, puffballs and molds, almost a poetic quality to these organisms. For anyone in the audience who has spent time eating anything with truffle in it, you know how delicious that is. For those who are really interested in strange, beautiful diseases, the cedar apple rust, shown with the orange and purple here, is a great example. When I was in college, I spent a little time working on a very interesting fungus called the anther smut. It's basically an STD, a sexually transmitted disease of plants, and it occurs in white campion back east. And it's super interesting in that pollinators move spores from flower to flower. The spores germinate, they grow into the plant tissue as hyphae. They then reduce the growth of female tissue. They induce the growth of anthers or male tissue. And then those anthers, instead of being full of pollen, are full of fungal spores, and they're ready to be transmitted again. Some of the fungi are spectacular and large, like our joyful puffball in the middle, our delicious morel over on the left, and some are tiny but mighty. So we see yeasts in the lower left, and over on the lower right is one of my personal favorites. That's a fungus we call a looby-dooby. It's the Labulbineomycetes, and that's a group that lives on exoskeletons of insects. They're super tiny, usually less than a millimeter in size. Really dark melanized structures. Nobody really knows what they do, but they can be incredibly specific, growing only on the shoulders of certain beetle species and certain river valleys. So there's a lot of beauty in the fungal world. Where do we find fungi broadly? Well, of course, everywhere is basically the short answer. Terrestrial, freshwater, and marine ecosystems. We can find them even in our own bodies. We are well known as bacterial habitats with microbiomes, but we also have our own mycobiome as well. And as we start to expand sort of the edge of human civilization outward, we carry with us fungal spores despite our best efforts. This is the Russian space station Mir, and it was home at its peak to several hundred species of fungi, definitely not space fungus waiting to invade Earth, but instead fungi that we had taken up there and then had allowed to flourish in the context of the space station. So fungi, of course, are extremely abundant in soil, air, and in fine art. I have this little gnome to remind me that there's a curious association between gnomes and mushrooms in art. And how abundant are fungi in air? Well, it depends on where you are, what the season is, and so on. But if everyone just pauses for a second, and something that's good to do in the evening anyway, take a deep meditative breath. Okay, and let it out. And so you've just passaged into your lungs and out again thousands of fungal spores. And it is remarkable that we are resistant to infection in so many cases. So the study of fungi, I think everyone will know, is mycology. It's actually a classic typo in that if you went back to the classical languages, the appropriate term should be mycetology, but we go with a little bit of contraction here and call ourselves mycologists. One last little background piece here when we're thinking about fungi is that fungi can be single-celled, like those little yeasts, the things that give us the holes in our bread and the bubbles in our beer. But most are multicellular. Generally, most species have a filamentous growth form. And there are a couple of just useful terms to know before we dive in. The first is that the fungal body as a whole is called the mycelium, and the plural is mycelia. And this one is easy for me to remember. Kirsten said that I am the curator of the Gilbertson Mycological Herbarium down here in Tucson, and that is correct. And the herbarium is named for a mycologist named Gill, Bob Gilbertson. For anyone who knows mushrooms have gills, you'll know a mycologist called Gill is really cute. And what's even sweeter is that late in life, he married a woman named Celia, he called her mycelia. So you had gill and mycelia, which is just about the most charming thing ever. So it makes it very easy to remember the fungal body being the mycelium. And it's made up of individual filamentous structures called hyphae. The singular of those is hypha. And so when we're seeing fruiting bodies sticking out of a log or growing up out of the earth, what we're seeing is the fruiting body in the tip of the fungal iceberg with a massive mycelium typically in the substrate where it's exuding enzymes and taking up nutrients. And if we zoom in on the mycelium, then we can see those hyphae. And the fungal mycelium, because it's based in hyphae, is really good at having an enormous surface area. So that's really important. Imagine that instead of a stomach and a mouth, you absorbed your food through your skin. So you would basically want to be in a bathtub of nutrients all the time. And that's what fungi do with all these hyphae. That surface area is so high because the hyphae are so small. So this thing that looks like a telephone pole is a human hair, and then you can see hyphal structures. They're typically about a micron in diameter next to it. Okay, so really small, tons of surface area, great for nutrient uptake. So on the one hand, that's awesome from the fungal perspective, but the challenge we have is that because fungi can spread rapidly, they can grow through many different substrates depending on the species, they can be very problematic. So our most costly plant pathogens are fungal pathogens. Here we see some pathogens occurring in grains and leafy greens. We see in chocolate trees, a very big problem in the New World. We see smut here on corn. And then here a colorized image of these fungal hyphae, looking sort of like some of the creatures out of the Moomin Troll books, if you remember those from ages ago. And so we see fungal pathogens not only occurring in cultivated plants, but also in natural forest ecosystems. And many of you will be familiar with those up there in Flagstaff. Fungi also can occasionally be pathogens of animals. And like I said, when we all breathed in those fungal spores, we were fortunate because we have many levels of resistance against fungal infection. However, there are emerging fungal pathogens that are a huge problem worldwide. And one example is Batrachochytrium dendrobatidis. And this is a really interesting group of fungi that is associated with amphibian decline worldwide. So frogs and salamanders are susceptible to a disease called chytridiomycosis. And it's thought that global change combined with stresses on animals for other reasons has led to these massive outbreaks that can cause enormous die-offs of amphibians in certain habitats. We do know, however, that some animals are more susceptible to fungi than vertebrates are. And this is an example. So this is a fly that's been infected with an entomophthora. That means literally the insect destroying fungus. And you'll see these around, especially late in the season. We're just sort of ending that time now, where insects will be infected. They'll be induced to have a behavior change. They climb way up onto a high surface or a screen on your home. And then the fungus bursts out as the insect dies, and a rain of spores comes out. This is one group of fungi that does this. There's actually another group that's converged on the same behavior. And that's where you may know about the zombie ants and that sort of thing. So when you're walking through a grassland or a prairie at this time of year, take a look around and you may see insects in that mortal pose. For those who occasionally get out of the Flagstaff area and might spend time under the sea, as it were, of course, marine fungi are an interesting area of discovery and unfortunately an interesting area of pathogens emerging right now. So this is an aspergillus that causes purple spot in corals. Considered to be a challenge in coral reefs. And then, of course, so many of our plants, when we're down here in southern Arizona, our agaves and columnar cacti rely on bats for pollination. We're fortunate not to have white nose syndrome here as an enormous problem in Arizona, but in other areas of our country, Geomyces destructans, a oft renamed fungus, is associated with bat mortality. However, if you do spend time in the southern third or 2/3 of the state, chances are you will have encountered the valley fever fungus. This is a group of fungi. There are a couple different species. Here in Arizona, we have one predominantly that moves frequently in our dry season following wet seasons. So these haboobs or dust storms sweeping over places like Phoenix are great sources of valley fever spores. So that's kind of the ugly side of fungi, but I mentioned that, of course, fungi are important for positive reasons too. And of course, perhaps the most important that we immediately come to on the human side is medicines. And so the apocryphal story of Alexander Fleming leaving plates, petri dishes in his lab and coming back and finding that a fungus had colonized them and was lysing or destroying bacteria, and thus led to, after the late 20s in the last century, the discovery of antibiotics, and penicillin being the first of those. So major antibiotics are, of course, incredibly important. We also, from fungi, have cholesterol-lowering drugs, anti-cancer medicines, and so on. So there's a tremendous wealth here. And when you think that there are somewhere between 1.5 million and 9 million species on the planet, most of which haven't even been identified yet or seen by human eyes, it's an amazing richness of not only biodiversity, but potential compounds as well. So as any coin has two heads, so too do fungi then flip and say, okay, well, we will make you lots of useful compounds, but we're going to take your strawberries. And so we know, of course, that fungi are major decayers of plant material. And if you're like me, those strawberries undergo this transition very rapidly in my fridge with these sugar-loving molds. They grow super fast and they love fruit. And so when we think of decaying plant material, sometimes this is of our food stuff, but most importantly, fungi are the primary degraders of plant cell wall material in nature. So they are out degrading fallen plant material. We'd be up to our necks very rapidly in plant material if we didn't have fungi, breaking things down and converting them into forms that are useful in soil. When you're walking around in the Flagstaff area, you'll see, of course, rotting logs, especially in this wet season. And what's sort of nice is that you can actually tell what broad functional group of fungi is in those logs based on what you see. So, for example, when you walk up to an aspen log that is nothing but these white, fibrous sort of leftovers, what you're seeing is a white rot fungus. And it is degrading the lignin, the really recalcitrant, tough material that makes up plant cell walls. And there are a bunch of assorted white rot fungi all over the place. Let me just see if I can hide my video panel, floating meeting controls. There we go. And so there are many white rot fungi that you'll see all over Arizona. Basically anywhere where it's wet enough and warm enough, they are happy. So here's a Phanerochaete. This is where we find oyster mushrooms also, delicious, especially when they're growing on hardwoods. And armillaria, so these actually are pathogens, and they cause a disease that is a white rot of forest trees. These photos I've borrowed from Scott Bates in his Arizona Mycota project. Okay. And so the other thing you might find, and we see this very frequently if you walk up to old pine trees, is you'll see brown rot. And in that situation, all you have are these sort of cubical pieces left over. And what's happened here is that the fungus has actually consumed cellulose, leaving the lignin behind. And so there are two really different groups of rotters. They have slightly different groups of enzymes and different approaches. A brown rot fungus that you might see is Phaeolus schweinitzii, typically on some underground material here. Fomitopsis pinicola is another example, here in all of its shiny glory. Unfortunately, this is where we also find quite a few of the saprotrophic fungi that can impact humans. So this is a photo that I borrowed from the internet, and this is showing a music room after the flooding due to Hurricane Katrina in New Orleans. Everything you see there on the formerly white walls is mold. So unfortunately, the ability of these fungi to break down plant material is problematic for us because we use a lot of plant material in our homes and habitats. So just to round out this sort of broad introduction to fungi, I'll just give a plug for the fungi that I study. So if you take a moment, please, everyone, your plant folks, so please picture your favorite plant, whatever that may be. I'm seeing thought bubbles above everyone's head, right? Thinking about plants. One of my favorite plants happens to be the chocolate tree. I worked on it as a graduate student, so it's got a special spot in my heart down in Panama. And so any of the plants that you have just pictured, in their above ground tissues and in their roots, they harbor groups of fungi called endophytes. These endophytic fungi are literally fungi that live inside plant materials without causing disease. So they live inside healthy leaves and healthy roots. And they occur in every plant species, and they're important because they protect plants against disease, against insects, against various stressors. And you would never know they're there because the leaves look perfectly healthy. But if you take those healthy leaves and you clean the surface so there's nothing on the outside, and you cut them into a little 1 millimeter by 1 millimeter piece and put them on a growth medium, an endophyte will grow out. And what's beautiful is that if you do this over and over again, you get these spectacular fungi, 95% of which are unknown to science, and all of which I think are as beautiful as a stained glass window in a church. And so this is where my research focuses, discovering these fungi and understanding what they do. And it ties in nicely with the history of our country, in that I think many of us are familiar with the Salem witch trials and the ways in which women were victimized because of purportedly aberrant behavior. And it's thought that in part that may, leaving aside the sexism of the age, may have reflected responses of the women to alkaloids, basically mind-altering metabolites from the endophytes that were present in the grain that they were grinding for bread. And so I'm interested in who are these fungi, what do they do, how did they get so darn beautiful, and how did they evolve to be friendly for plants? And so my lab and I work basically from the Arctic to the tropics on questions like that. And one of those questions has to do with how the endophytes protect plants. And so here I just want to show you one more little chocolate tree. And this one is showing us that you can actually raise plants without endophytes because they accumulate as the plants mature. And in this case, we raised plants without endophytes. Then we put in endophytes, and then we added a pathogen. And what we see is that when you've got the endophytes there, the pathogen doesn't really get a foothold. But when there are no endophytes, that pathogen can really do some damage. And so there's a lot of future in local sustainable chocolate using endophytes, an area of excitement for me. Now, it turns out that many of us also then are familiar with lichens, right? So as you're walking around the beautiful rocks up there in northern Arizona, you see a diversity of lichens. And then when you get up into the hills and you've got mosses with these beautiful Peltigera lichens, this is another place where fungi are super common today for all of us to see. And they're nice because they often persist for many seasons, growing slowly in these environments. Now, typically, each lichen consists, of course, of a fungus, but also a photosynthetic partner that's either a bacterium, cyanobacterium, or an alga, or both. It turns out that these also are home to many endophytes. The endophytes live with that photosynthetic partner, so that's another area of interest for me. So when I go out and see beautiful spreads of lichens, I'm thinking, wow, that's an amazingly successful fungus living in symbiosis with a partner and providing a home for a diversity of additional fungi, many of which are not yet known. Okay. So just a few beautiful lichens. This is a nephroma, and this is an especially cool one. It's growing here with a moss, a little pleurozium moss. And in this case, these dark spots are little pockets of cyanobacteria where they can fix nitrogen, atmospheric nitrogen. And then here in Arizona, you'll see this one very frequently. This is a Peltigera, and it's really common, for example, on the slopes as you are walking up, if you're climbing Mount Humphreys, for example, in the Kachina Peaks Wilderness. It grows on mosses, typically in slightly sunny little banks right on the trail. It's really beautiful. It's a beautiful lichen. And some of our species here in Arizona have this very pretty surface called a tomentum. It's kind of powdery, and it provides some protection against environmental stress. Okay. Last but not least, of course, when we're talking about fungi, we've talked about how they break down plant material, and that's super important for soils. We talked about how the ones in leaves and roots can be helpful as endophytes, protecting against disease. And then another super important thing that fungi do is, of course, enhance the uptake of nutrients from soil by forming mycorrhizal symbioses. Up there, when you're looking at the beautiful ponderosa pine forests, for example, and if you imagine just a little tiny pine seedling with a little tiny root area, surrounding that root area is the extent, that massive surface area of the ectomycorrhizal fungus that, in association with the roots, modifies them to these club-like structures with very distinctive morphology, and is really great at helping these plants take up the nutrients that they need. So pine trees and aspens and a variety of others have these ectomycorrhizal fungi. And then the vast majority of plants, at least 80% of land plants, have endomycorrhizae, which we now refer to as arbuscular mycorrhizae. And these are inside roots. And we see these in things like poppies, saguaro cactus, and so on. Together, all of these mycorrhizal fungi are super important in nutrient cycling. They help with nutrient uptake, water uptake, and resistance against disease. And so often people say, oh my gosh, like, how do you study fungi and live in a place like Tucson? Well, as I'll show you in a moment, we are at a threshold of beautiful fungal diversity here. And many of our fungi are just more subtle. So when I look at this beautiful spot in the Tucson Mountains, one of my favorite hiking locations in the saguaro, I think, there are diverse arbuscular or endomycorrhizal fungi, diverse endophytes, and even some macrofungi here that are worth some attention. Okay. All right. So when we are thinking of fungi, though, the vast majority of us think of these beautiful macrofungi, mostly basidiomycetes and ascomycetes. And when we walk into a forest, it behooves us to think of this beautiful structure that is below the soil surface beneath our feet, and to understand that those fungi are incredibly vital for the health of the forests. There are some really great experts in mycorrhizal fungi up there at NAU, and I'll just mention Nancy Johnson, who works on arbuscular mycorrhizal fungi, and Catherine or Kitty Gearing, who is an expert in these ectomycorrhizal fungi. So really great scientists and wonderful people to talk to. So what I'd like to do now is just transition to share with you some of the beautiful fungal diversity that we see here in Arizona. And in doing so, we're going to take a little elevational gradient walk. And we're going to start in my neighborhood here in central Tucson. So I live about a mile from the U of A campus. We are surrounded by a large number of non-native plants, but occasionally we are fortunate to find fungi that are native here and are truly spectacular. So let me just introduce you to a few of them. So this is Batarea. Batarea is a stalked puffball, and it grows beneath mesquite, but also beneath things like Aleppo pine and some eucalypts. When these fungi first emerge, they're saprotrophs, so they are breaking down plant material to make soil richer. They look like ping pong balls on a stick. They can be up to 2 feet tall, so they're really dramatic. Here we have a cyclist in the background for scale. These are about 6 inches tall. And that ping pong ball ruptures, and then there is a spore cloud inside. Millions of spores. And so these sometimes rupture to look like salt shakers. In other cases, the top just sort of breaks off like this. This illustrates something that we see very frequently in desert fungi. Because our environment is so dry, the glorious macrofungi that can flourish in a wet environment don't do as well here, and they need strategies to protect their reproductive structures during the short wet periods that we have. So Batarea is an example of a puffball on a stick. It has a shell until it's ready to let those spores out. It's protected inside that shell. Another example is our desert stalked puffball, something you'll see all over Tucson and Phoenix. This is Podaxis pistillaris, and it often grows in these little troops. These guys also about 4 or 5 inches high, up to about 10 inches tall. And they are super common actually in the landscaping that's been done all around Tucson on our bike path. So mulch and other substrates have been brought in. You don't see any plants coming up there in the rainy season, but you see hundreds and hundreds of these fungi. These also are basically a tightly closed shell that ruptures, and then black spores come out and drift away on the wind, making more of themselves. We do get some mushrooms in well-watered environments, and this is the most infamous. So this is Chlorophyllum molybdites, and it's the #1 cause of mushroom poisoning in North America. It's not deadly in most cases, but it does cause gastrointestinal distress. And often people mistake it for an edible lepiota if they're coming from, or macrolepiota, if they're coming from the Midwest or back east. The giveaway is that these gills, you can see the tightly spaced gills here, are kind of an olive green color. Unfortunately, sometimes people are eating them at this stage where the gills are not evident, but we've gotten pretty good at being able to tell what these are for all those poison control calls that we get. Okay. They grow to the size very rapidly, and they form fairy rings in our Bermuda grass lawns in southern Arizona when there's lots of water. Perhaps a stinky one to mention, but a good one, is Itajaya. This is a stinkhorn. It exists as these sort of egg-like structures underground. Sometimes people contact me wanting to know what reptile has laid eggs in their yard. But if you slice them open, they're gelatinous and slimy inside. And the emergent mushroom smells like rotting meat. It is a distinctive smell. I can pick them up usually at about 10 feet on a good sultry monsoon day. They are visited by flies that are attracted by the terrible smell, who then move the spores around. These are very common under mesquite and eucalyptus down here in Tucson. Now, as we start to go up in elevation a little bit, one of my favorite field sites is just south of Tucson, the grasslands down around Green Valley and Sahuarita in the Santa Rita Experimental Range. And they share some traits in common with the grasslands of central Arizona, like Chino Valley and so on. And in this area, you've got mesquite trees or other chaparral type trees, and you'll start to see some familiar fungi like Leucocaprinus here. And this is the land of the puffball. So these are still arid environments, although it's about as lush as it gets. This is the Santa Rita Mountains at my field site, about 45 minutes south of Tucson. And I'm standing in a sea of non-native grass. This is all layman's love grass with mesquites in the background. And you can see that many of the fungi here have that same strategy, a closed structure with their spores inside. And so there's lots of Bovista puffballs there. Now continue up just a little bit, and all of a sudden we're into moist enough microhabitats with oaks. And here we're seeing the beautiful shield lichens in the Parmeliaceae. And some of the most delectable edibles in my book, this is the lion's mane mushroom. This is a picture that I took earlier this month down in the Santa Rita Mountains. And we'd come up now out of the grasslands into the oaks, and lion's mane was literally everywhere. So we collected some for science and some for food at our field site there. Really spectacular. These are slow pathogens on the trees, but they are also very strongly antimicrobials. So you can pick these and snack on them for long periods of time. They're also unmistakable. Nothing else looks like a koosh ball and cousin it got together and climbed up into a tree and made a beautiful baby. This is where we also see earth stars, and I'm fond of these. I grew up in a family that didn't have a background in the sciences, but I was fortunate to spend time in the chaparral, oak, and pine forests around Prescott. This was the first thing that I found that I was able to look up and identify as a fungus. I didn't have anyone to tell me what it was. So these are the water measuring earth stars. This is after our first monsoon rain this summer, and they've deployed their little octopus legs. They slowly dry up and curl up, and then they'll open again when conditions are wet. So these are fully open, and then the spore bag in the middle is a puffball that upon being touched or poked or stepped on or rained on, then will be spewing spores out. So these are really common in sort of the oak, juniper, woodland areas right on the soil surface. And they're really fun for kids to find. Kids love them for some reason. I do too. So I mentioned that once we're in wet enough environments, higher elevations in Arizona and riparian areas, fallen wood stays wet enough to support diverse fruiting bodies. And this is where we find things like turkey tail mushrooms, Asterium trametes. We also find Daldinia. This is an ascomycete that if you cut open has concentric rings inside. It's really distinctive, slowly but surely breaking down that old wood. We also find additional ascomycetes like these beautiful cup fungi, and these can be very subtle. These are less than about ½ inch in diameter down in the leaf litter, and you can see that I'm in sort of a pine fir forest again in the Santa Ritas. This is a picture from last month when it was so wet up there, really fabulous. These are saprotrophs. And then many of you may have seen this particular mushroom. This is a mushroom in the genus Helvella. It's an ascomycete as well, also really good at breaking down plant material in soil. And here in the Santa Ritas, it was growing at about 5,000 feet. It's meaty enough, it was a handful. And these are often poisonous. They produce compounds that are similar in structure to jet fuel, so pretty dramatic. I always think they look like they're straight out of Harry Potter. They're kind of creepy skeletal things, super neat. So you all up in the 6,000 to 8,000 foot range, sweet spot around 7,000 feet, are in a fabulous place for mushrooms. This is from several years ago, a foray that we led with students on Mount Lemmon at about that elevation here around Tucson. And at the height of the season, I think you know, it's really spectacular to be in our western forests for the mushroom fruiting. It is joyful. The diversity of things that you can find in just a small amount of time is truly spectacular, spanning literally millions, hundreds of millions of years of evolution. And so every mycologist like me gets very happy. We start to see Sporacis, a beautiful cauliflower mushroom here. And we also see things like Ganoderma. The challenge, of course, is that our sky islands, our forests are changing. They're drying over many years with prolonged drought. And of course, when we think of the impacts of fire, we recognize that fire will produce fruit flushes of certain fungi, but in our changing climate, it is harder and harder for many of these fungal communities to reestablish. So we always want to just bear in mind that collections we can make today may not be possible tomorrow. And it's one of the reasons that when we collect, we try to do so both with great joy, but also with great responsibility. In these forests, we'll find oyster mushrooms. We'll find the autodeliquescing mushrooms, like the inky caps. These are interesting in that they typically are not problematic for many people. However, some people will have toxicity effects if they've drunk alcohol within 72 hours of consuming them. Of course, you don't want to eat them when they're super drippy like this, a lot fresher. So that's the Caprinus or Coprinellus group. The oyster mushrooms, like I said, tend to be much more delicious here when they grow on hardwoods. They get a little bit tangy when they're on conifers. The beautiful Ganodermas that we see that are these polished mushroom polypore structures, shelf fungi, are really worth it. I'm a fan of the subtle little fungi that you find by crawling on your hands and knees. So this is Claveridelphus. They're about the size of matchsticks, and they grow under Douglas fir in the duff. And they're little saprotrophs, often this sort of yellowish color, really compelling and beautiful little fungi. And of course, we can find things like coral fungi in our forests, and then even tiny little bird's nest fungi. And you'll find these in potted plants as well. And in this case, they've got this little membrane that ruptures, and you've got these packets that contain spores that splash out with rain. These are less than a quarter of an inch in diameter, so really beautiful, and I love their furry little cups. Last but not least, we'll just end on a few favorites. I'm a huge fan of puffballs, like lion's mane. They're delicious, dry sauteed with a little bit of butter, a little white wine maybe. And here with puffballs, you just always want to slice them in half and ensure that you're not seeing the outline of a mushroom in there, because the button stage of toxic amanitas can look similar depending on the species. Here's another example. So these are puffballs that are common around the Prescott area, and really, really nice. Okay. So when we are collecting, we are doing it largely for science. And this is what my office looked like after our most recent foray. We'll just do a flyby of my desk. We collected in about 6 hours of surveys for biodiversity work. There's our lion's mane. Over 75 species. And it reminds me to say that the vast majority of areas in Arizona have not been thoroughly sampled for their fungi. So we have all of these individually wrapped in wax paper. And for each, we've gone through a process of collecting data in the field. We're now preparing to photograph and dry all of these specimens, and then we're placing them into the Gilbertson Mycological Herbarium so that we have a permanent record of that biodiversity. And we'll talk about the herbarium in just a moment. Before I get there, I just wanted to introduce a new project that we're working on. And the reason we just did the elevational gradient is that that is part of this new project here based at the University of Arizona, and it's funded by the National Science Foundation. It's a project looking at how climate influences fungal biodiversity. And I'll just give you the 50,000-foot view here. Our site is in southern Arizona. We have sites in the project in Alaska, in Oregon, Colorado, in Kansas, Minnesota, Florida, and in the Northeast. And in each of these are long-term research sites that have a wealth of data. We're going in and wherever possible, sampling the macrofungi, the mushrooms and bracket fungi that we find in pine-dominated or Pinaceae-dominated forests, in oak woodlands, and in grasslands. And so very much like the little elevational gradient that we just did. And in each of those, we're looking at sites that have been burned or not burned over time. We are going out with community scientists to look at what macrofungi we can find at the sexual reproductive stage, the mushrooms and bracket. But we're also collecting leaves for endophytes, roots for endophytes, spores in the environment, and soil fungi. And our goal eventually is to understand how climate and fire in each of these different biological plant communities influences the distribution of mushrooms at this national scale. And so I mentioned this in part because we are partnering with the experts in fungal diversity in Arizona, and that is the Arizona Mushroom Society. These folks know their stuff, and they are awesome. And starting this month, in just a couple of weeks, so on October 6th, we're going to be offering our first DNA workshop here in Tucson. We'll have another one on a Saturday following, and I'll share these with Kirsten and Sue after the meeting for anyone who would like to come down to Tucson and learn how do you go from a mushroom to a DNA sequence. And then we also will be hosting forays next summer in the grasslands, pine forests, and oak forests in the Santa Rita area down here. Last but not least, I wanted to mention that the Mycological Society of America, which is the National Society for Fungi, is having its national meeting in Flagstaff next summer. And so we're really excited. We've never had an MSA meeting in Arizona before. And so there will be forays and educational events associated with that. And we look forward to sharing information about that. I am honored to be the immediate past president of the MSA, the Mycological Society, and to be working now with the leaders of the Arizona Mushroom Society on these new projects. Okay, so in our last couple minutes here, I just want to share that one of the hats that I wear in addition to doing research and teaching is that I am curator of the Gilbertson Mycological Herbarium. I have included our website here, but you can find it if you just search Gilbertson Herbarium. And this is the preeminent collection of fungal biodiversity in the Southwest. We're really proud of it. I joined the faculty in 2005 and am proud to curate this classic collection. So our current holdings are about 42,000 accessioned specimens. So these are specimens with labels and data in a box. This includes things that are fungi and then fungus-like organisms, a few photos here. And then we've also added almost 200,000 endophytes to this. Our 42,000 number is growing now through our climate mushroom project and through partnerships with Arizona Mushroom Society. So that's really wonderful. Our cabinets are filled and you can find these labels, some of which go as far back as the 1800s. And it's especially powerful because we can get DNA from that material. We know when fungi were fruiting back then, so we can look at changes over time. And we also can get a sense of how biodiversity has changed over time. So it's a very useful collection, and I use it every time I get a poison control phone call. So this is what one of our specimens might look like. Okay. We also have a spectacular photographic record, and I love showing this to my college students because they're not exactly sure what technology they're seeing here. But of course, our old photographic slides, some glass mounted, some cardboard mounted, and with tons of data. So I've had students help me basically transcribe and then scan all this information so that it's available as a teaching resource. Our collection includes spore prints, super important for identifying a number of fungi. And then finally, we provide resources for those who are interested in learning how to collect fungi. This photo again comes from Scott Bates, and this is from his Arizona Mycota project. What sorts of things might you like to take in the field when you're going out to collect some fungi? You've got your wax paper. We never want to put our fungi in plastic bags. You've got little tackle boxes or craft boxes to keep your specimens separate. You've got a brush to get that dirt off. You've got a notebook. Now you've got your phone, the world's best camera, a trowel or a knife, and a hand lens, and some guidebooks. And with this and a nice mushroom basket, life is great. And our NSF grant has now generated a field collecting protocol for macrofungi that I'll be happy to share again with our organizers for tonight, and they can distribute. And this just talks about the best practices. If you want to collect specimens and identify them and make them part of the scientific record, this is a super important role that community scientists can play. We can't be everywhere, and community scientists have a great ability to find things that we don't find as professional mycologists. And in fact, I don't even like that term amateur mycologist. I feel like everyone is a professional or paraprofessional. Basically, to spare you some reading, the cliff notes for this document are basically take enough, take it carefully, take a photo, and take the data. And that's what we're looking for. And by a quality collection, we mean collecting the whole mushroom. We are using INAT for our field collections in this project with all of our community scientists, and it's working great. What's awesome about INAT is you don't have to know what it is. You can post it, other people can help identify, and you might get some great suggestions for what you might be seeing. And then I love this one. This is the heavy labor of curation where you're like, okay, now I have to record all this information. One simple rule that we have is to dig, not pick. So when you're coming up to a little mushroom, instead of plucking it, dig it out, because often there's structure underground that's really important. And then we ask all of our collectors to just bear in mind that you want to keep your specimens intact, not crushed, and try to keep them separate from each other by type and locality. And then we dry them and include notes about them, and then move that information into our database. So this is just a quick snapshot of my INAT page, and you can see that sometimes I know what I'm looking at, sometimes I don't. That's okay, but getting those records up there means that you've got a permanent biodiversity record. You are contributing to the world, and I highly recommend it. They're becoming more and more useful for scientists and for people who are just interested to learn. And then our database goes to the MICO portal. In our herbarium, we database all of our records, and they go to this national web accessible database. You can actually generate like collection checklists, what fungi are known from Sunset Crater, and get that list and then take that out with you. And I guarantee you'll find stuff you've never seen on that list, and that's an important observation to put up in INAT. And so if you go to our herbarium database there, you'll see that we, when I grabbed this snapshot, had 26,000 specimen records in there, the majority of them identified to species, representing over 3,000 species. And that's about how many known fungi are in Arizona, but the true number is going to be much, much greater. Okay. And so with that, the last thing that I'll say is that I serve as the poison control contact for Southern Arizona and low elevation Arizona in parallel with Dr. Kitty Gehring at NAU. And so when people, for whatever reason, ingest mushrooms and are unsure what they have consumed, we get the phone call from poison control. And our herbarium is a great reference for that, because we have fungi just like the chlorophyllum. We have young ones, mature ones. We can really do a lot of identification. Most mushrooms in Arizona are not poisonous, but we've got some real doozies, and it's important to never consume them without absolute certainty. So that's my plug for please don't eat the mushrooms unless you know what you're doing. There are a lot of great references out there, and our herbarium does do identification, but typically will not make recommendations about edibility for liability reasons. Okay. Okay. So last but not least, we partnered with Poison Control to come up with the sorts of questions that we need the public to be able to answer when people do ingest mushrooms. And we try to make this appropriate for a parent who is freaking out because their toddler is chewing on something from the yard, or a college student who's totally not sure what they thought they ate up on the mountain. And so they're very straightforward questions, but we've found that this works really well in getting us a lot of information. How big is it? Do you see a ring? What color? From above, from below? Base of the stem, does it have a smell? These really help us dial in what we're looking at. Sometimes we get photos like this, and we have a pretty hard time guessing what the person ate. Sometimes we get photos like this, and it's like, oh, absolutely, 2 different life stages, below ground, above ground, distinctive features, A+, no problem. I'm not sure who would want to eat that. That's a super nasty stinkhorn. Sometimes we get habitat, size and color of the cap. That's helpful. Okay, sometimes we get measurements, but unfortunately looking at the top is not super helpful. Sometimes we can see enough to guess about gill attachment, size and color, size. And then occasionally we get a beautiful photo that shows us the entire fruiting body along with answers to all of our questions. And when we get that, it's an A+, and we can identify with certainty. And this is an example of one of those chlorophyllum common poisoning mushrooms that we had enough information based on the gills, the base, the top of the cap, and the size. Okay. So that's just a little bit about what we do here down at the U of A. I love our beautiful state of Arizona. I think our plants and fungi are spectacular. We're at the confluence of multiple biotic provinces that gives us a tremendous richness. And I would welcome any follow-up questions you all may have. I can be reached here at the U of A, and it turns out that if you Google Betsy Fungi Arizona, you can find me. And hopefully that gives you just an overview of why things here in Arizona are so cool from a mycological perspective. And I really just want to thank you again for having me tonight. It really was a pleasure to share this with you. So thank you. Thank you so much.
Thank you very much, Betsy. That was a marvelous presentation and very fascinating. I'm going to go ahead and stop the recording, and then we will open it up for questions and answers.
Great.